we thought we would try to be more wet than you. This is the first. I've never given a talk this soggy. But we're really excited to give an update about this collaborative project in the Western Lake Erie Basin. This is funded by the Michigan Department of Agriculture and Rural Development. We're going to discuss some early results today. So just say that right out of the gate. This is very preliminary, but also some exciting initial findings. And we also want to have plenty of time to hear your feedback on what we've done so far and next steps. So I'm going to start just with the overview of the project team. I'm Jennifer Blesh from the University of Michigan, co-presenting with
Brendan O'Neill. Allison covered it.
Yeah, I think Allison covered our backgrounds. We're partnering with Vincent Denef. He's a microbiologist at U of M with MSU's Institute of Water Research. You've already heard a lot about those complementary projects today. Christine Sprunger at Kellogg Biological Station. Julie Dahl at Michigan Agriculture Advancement. Angela Blatt, Alliance for Great Lakes, and also the Michigan Climate Smart Farms Project. So our overall goal with this work is to partner with farmers in the basin to advance knowledge about this relationship between improvements in soil health and changes in water quality, while also supporting adoption of regenerative practices on farms in the region. And we're really thinking about an influence on adoption in a very multi-scaled and holistic way. So one way is this feedback that you see here in blue, that at the individual scale, there is a relationship. There's this potential for adaptive management as farmers experience changes in soil health through time that can improve yields or yield stability or reduce input costs that can lead to a positive feedback that ultimately supports water quality. We also recognize that big changes in management systems are really hard to make because of policy and market structures. We have no illusions about that. And so one of our goals is to really build the science base to help inform shifting incentives to where they're most effective. So Brendan's going to briefly define soil health for us. And I also was just going to say we're going to sort of jump back and forth in this talk, partly to keep you awake on a Thursday afternoon.
I think the rain did that, but. Okay, so soil health. For really decades at this point, it's hard to believe that. Soil health has been defined as sort of the capacity of a soil to function as a vital and living ecosystem. But just like the soil health sort of knowledge and testing is progressing, so is sort of the definition of soil health. And so a more kind of updated version that I've been using is thinking about soil health as soils that retain functionality, vitality, and sustainability or resilience. So functionality in the sense not only of utility to agriculture, but within the context of the broader landscape or broader ecosystem, that they support healthy ecosystems. Vitality, soil isn't just a collection of components, right? Sand, silt, organic matter. It's truly integrated and has interconnected processes, many of which are mediated by microbes. And we're trying to get at that a little bit in our research. And that sort of vitality is often, it's not necessarily tangible, but when you see it in the soil, you can say, oh, this is good soil. And then finally, there's an important time component that I think is important, especially for this meeting. You just saw all these, changes, seasonal changes in total loads and flows. Same thing with soil health. It's a snapshot in time, it's region specific, and our definition for one region or one time to another may evolve.
Let's see if it's possible to move this here so our heads aren't part of the display. All right, that's a little better. I don't have the fun shadow there. Okay. I'm just going to briefly, this looks really complex. I'm an ecologist. I love diagrams like this because an important way to improve soil health is to apply an ecosystem approach to thinking about nutrient management. Farms are ecosystems, and they have all of this complexity governing nutrient flows and cycling in the soil. And so we really want to think about not just managing this one small little flux here of fertilizer, but managing all of the ways that nutrients are cycled and supplied to crops and stored or potentially lost from soil. And so we're thinking about all of these microbial processes like decomposition, assimilation that help to supply and recycle nutrients. legume nitrogen fixation as a source of nutrients. So I just want to keep in mind that we're thinking about the relationship between soil health and building up these multiple fractions of organic matter and how that relates to how nutrients are cycled and stored in soil. And so when farmers use practices that increase crop diversity, for example, increase microbial activity, you're really ramping up these other mechanisms that supply nutrients to plants and can ideally reduce the need for external inputs. So this study site is in the Michigan's portion of the Western Lake Erie Basin. Our partners mainly came from Lenawee, Hillsdale, and Monroe, some fields in Washtenaw County, so you can just see the big picture there. And then here's a more detailed breakdown. We've actually continued adding more fields to the study since I first made this slide. I think we're up to 66 or more row crop fields that are participating. We also have 21 fields that are in specialty crops, so diversified vegetable or livestock, that were recruited through the Michigan Climate Smart Farm Project. And then we also sampled 10 woodlots. So farms that had unmanaged areas that were adjacent to cropped fields and on the same soil type, just to help us have a better sense of reference levels of soil health for particular soil types within this region. For our approach, most farms decided to enroll 2 fields in the study, so that allowed us to have often a nice paired approach like you see in this picture here, 2 fields side by side, maybe similar soil types with different management histories, or sometimes farmers pick soils with really different soil types to understand why they perform in different ways. And then we selected fields that had a pretty stable management history, because this is an observational approach, and we want to be able to link management practices to soil health. So to do that, it can't be like, oh, I implemented this practice yesterday, right? We need to have some history of that practice being in place. So farmers enrolled fields that had pretty stable management histories for the past 5 or so years. We conducted in-person interviews with all of the participants this past winter to collect detailed information on those management histories, everything from crop rotation to yield to nutrient inputs, irrigation, and tillage, and sometimes going back way further, historical land use. I think I missed one. I'll just do this briefly. I just mentioned that one goal of the study, you just heard about MNRD's new regenerative agriculture program. We sampled fields in our study to capture the full spectrum of practices that are present on the landscape, because we want to relate management to soil health. So this full continuum here of different degrees of tillage intensity, different levels of crop diversity, different degrees of dependence on external inputs. But we intentionally oversampled a little bit on the more regenerative end of the spectrum. Those practices tend to be more rare, but we really wanted to build the database on how they influence soil health and potential water quality outcomes. So we paid particular attention to practices like reduced or no tillage, use of cover crops, cover crop mixtures, perennials, and integration of crops and livestock. And this is because we have the emphasis on these particular practices builds on ecological science and literature, showing that they have known mechanisms that restore organic matter and the many functions that it provides. So we're going to shift gears to showing some preliminary results from what we're calling the first phase of the study. So that's this observational piece that's currently underway, where we sampled all of those fields in fall of 2024, most of them right after harvest, depending on what crop was in the ground. And our goal going forward is to be able to pick out soil health indicators that really are sensitive and appropriate to our region, and that tell us about the potential for nutrient conservation or accrual of soil organic matter. So I'm going to turn it back over to Brendan to explain the soil health approach and some of those early results.
So what are we measuring? I'm hoping we have kind of a broad audience here in terms of backgrounds. And so I want to go over what a soil health test is and how we're going about it. It might be a little bit unique or specialized for the region. So on the left-hand side, you can see broadly what we use for biological indicators of soil health. So the amount of nitrogen that's supplied, that's not mineral nitrogen, let's say supplied by the farmer. Several fractions of organic matter, so mineral carbon, something we call POC, free particulate organic matter, Sometimes we know exactly what those fractions of carbon are and maybe the role that they play, like particulate organic matter. Sometimes we don't necessarily know what the fraction is, but we know that it's sensitive to management. And so over time, they've sort of been integrated into the test because they're responsive to changes in tillage or changes in rotation. Sometimes these organic matter fractions indicate something about the habitat, again, thinking about that interconnectedness of the soil. And so we can think of habitat as something like aggregation, or it might indicate something like carbon that's available for the microbes to really get going and sort of supply that engine of microbial activity. The great thing about this work is that we can link common measures that are used in soil health tests with some of our other approaches that are sort of research-based, but also can hopefully be on the leading edge of new soil testing methods and new soil health methods. So we measure enzyme activity, microbial biomass. We also extract DNA so we can understand what the microbial populations are and how they're associated with soil health. And we look at the food web through analyzing the nematode community. Then chemical indicators are broadly carry over from sort of long histories of soil testing and are integrated into soil health testing. In particular, we're looking at how we can analyze phosphorus in sort of novel ways given the importance of this region. And not just using the traditional method, which is Bray 1, but a modified method that helps us get at a lot of different fractions. And then physical indicators, many of which are the type of thing you can walk into the field and just know. Compaction, sandy, crumbly, that all are critical to soil health. What's that?
You're still up. Yeah. Although maybe one thing to mention here is just that a lot of those are still in progress.
Oh, yeah, you'll see some of these results, but some of the DNA tests, some of the enzymes are done, but we're not ready for showtime. Some of the DNA analysis we don't have ready yet, but a lot are completed, and this was the first phase of getting them done. So these are 3 different types of sort of phases of how we sampled in the field. So on the left, you can see soil sampling to 20 centimeter depth. We took multiple samples across field types, and we differentiated fields by soil type. So we went into the field knowing what the different soil types were. We collected broadly a composite sample across each field type, which we use for then separate in the lab and use for downstream analysis. In the center here, we took bulk density measures, so sort of a cylinder of soil, and whether a soil is compacted or whether it's well aggregated and sort of fluffy, we can use that to understand the concentration of carbon, nitrogen, phosphorus that are in the soil. So it's really critical for understanding the abundances at depth and across the field. And then finally, penetrometer readings. We took a lot of them all over these fields, over 3,500, to understand the compaction of the soils and the penetration. So we successfully captured, I think, our goal of sampling a wide range of different management practices in the study. So we had a lot of fields that were in cover crop, that were shown here, for example, but also a lot of fields that were maybe had a long-term history of no-till, were frequently tilled, were in a CRP program or some other long-term conservation program, some that had been rotationally grazed, some that received manure or compost additions in the past, and fields within nitrogen and phosphorus management practices, including VERT, side dressing, and reduced application rates. So here's another nice image of a cover crop mixture, really dense and lush in the fall. And just another contrasting example of soybean residue in one field, and then off to the right, an alfalfa field, both of which were sampled. And then this is an example of the forest plots that we measured. We used the same approach. Basically, we have 10 forest plots, as Jen mentioned. They're forests, but really they're often just woodlots that are associated with the farm and often adjacent to fields that we're already sampling, so they act as sort of a reference. And they give us an idea of how we can interpret soil health from that reference, right? Because we know that soils in less disturbed soils will maybe have different horizons of carbon. Maybe our measurements will, it'll indicate something about the past history and the way that those soils are acting in relation to other soil health parameters. And finally, some images of some of the climate smart farms that are in the program. Generally much smaller farms, vegetable farms, much more diverse, lots, including plant and animal diversity. And then just an example across the landscape of types of fields that we are, we're able to sample. So neighboring farms, for example, that have rye or alfalfa after corn or after soy, or and exhibit sort of the management gradient that we're trying for across the Western Lake Erie Basin. And I should mention also this was led mostly by, this enormous effort was led by Beth Van Duzen, who clearly from the pictures was also in charge of taking the pictures because she's not, somehow does not appear in them, but she was in charge of this. So, some results. So on the top we have some of those measures of biological soil health. So mineralizable nitrogen, this just shows the range of values that we captured across our gradient. Particulate organic matter, again, a range. There we go. Epoxy, this is one of these measures that we know is really responsive to management, but it's not totally clear what we're necessarily measuring in terms of the carbon pool, but it's been a very useful soil health measurement. And then some examples of the range of physical measures. So bulk density, percent clay, so we have a nice big range of clay content, and available water capacity, or how much water is retained in the field after at field capacity. So now that we have this nice big range, we can break down how some of these differences in the gradient that we're interested in, how they look. So in this case, we have fields with different histories of cover cropping, either a history of, over the past 5 years, of over 50% of the time having some sort of cover crop, or less than 50% of the time having some sort of cover crop. So in this case, this available water capacity, there's a clear difference with greater use of cover in the available water capacity that a field has. And similarly with this mineralizable N, this is again pools of N that come from organic matter that kind of are like a slow drip of nitrogen throughout the season as organic matter is broken down, is much higher under our cover crop sampled fields than the no cover crop. And then we have a few interesting phosphorus results so far. So this is sort of the traditional gray 1 phosphorus that I pointed out earlier. This would traditionally be used in a soil health test, but we know, especially now with some of this data, that we can do a better job. So this is no-till concentration of gray phosphorus versus tillage. So there's something going on with the till versus no-till and how we're capturing phosphorus levels, or maybe within management. However, just one frame of reference is that there's no difference in yield for till or no-till, even though we have these striking differences in phosphorus that we're picking up in the test. And really the numbers of tilled and no-tilled in this case are basically split half and half. So it's not some effect of some unusual farms.
I'm going to just point out a few things with the comparison with the woodlot. So in this case, it's not just pairing those 10 fields that were side by side, but this is the mean of all of the row crop fields and then the mean of the woodlots. And this is probably very expected results, but putting it here just for reference. So on average, soil organic matter is about twice as high in the reference woodlots, and this is sort of common knowledge. You see this cited a lot, that with the conversion to agriculture, we tend to lose about half of the original organic matter. One thing that's interesting to me, so in this project, we're really interested again in these different fractions of organic matter. These are measurable fractions that correspond with known different functions. So particulate organic matter is more recently derived organic matter, coming a lot from like plant litter or even manure inputs, and it turns over relatively quickly. So this is a small fraction that's really important for nutrient supply to crops. And that was about 3 times as high in the woodlot, which is interesting, and also made sense because woodlots have a lot of like litter carbon coming in, right? Leaf litter. And then mineral-associated organic matter we really think of as being important for long-term carbon stabilization in soil. It turns over more slowly, on the order of like decades to hundreds of years. And so we definitely want to build up that fraction too, thinking about climate mitigation and resilience and just all of the physical functions that organic matter provides. And that tracks with this organic matter number, about twice as high in the woodlot. And then we also looked at some comparisons with these soil health indicators that are available in commercial soil health testing labs, and also see some of those same differences. So as Brendan mentioned, POC is just sort of this quick, high throughput method that's a chemical procedure that seems to align probably with like that mineral associated organic matter fraction, more stable carbon, so also higher in the woodlot. And then again, you can see mineralizable nitrogen about 3 times as high in the woodlot. And so these are useful just for thinking about like levels to which we might aspire as you're really, never going to achieve this in a disturbed annual system, but can certainly get closer to the reference. As Brendan mentioned, we had some interesting things we were thinking about with phosphorus. Unfortunately, we don't yet have the more interesting phosphorus data yet, but I think this on the left here is sort of known, but also I just wanted to put it up here to point out one way that we're thinking about this. So as clay increased across our fields, we get into heavier clay soils, this measure of plant available phosphorus decreased. And that's sort of a known thing, right? Farmers on heavy clay soils think about how they're challenging to manage. There's a lot of mineral sites that can quickly pull phosphorus out of solution and make it unavailable. That's a challenge. But we also know that when those soils have a lot of rhizosphere, a lot of root activity, a lot of living cover, healthy microbial communities, there are mechanisms through which plants and fungi can access some of that phosphorus that's found in minerals and solubilize and mobilize that phosphorus. So especially in our fields that are in more regenerative practices, this is probably not telling us the full picture. And I put that graph there on the right to help illustrate that too, because 3.1P is exactly the same in our woodlots and our cropped fields, even though I just showed you that soil health is about twice as high in the woodlots. So we really need to do a better job of indicators of phosphorus availability through the lens of soil health and soil health testing. And so in this project, we're trying that through a multi-pronged approach, and Brendan's going to tell you a little bit more about that.
Okay, you asked for it and you came through the rain. So one more complicated picture, but this gives you an idea of where we're trying to go from some of these simpler and cheaper, quick measures of soil health to trying to understand some of the mechanisms at play. So I'll just run through this at a high level. So here on this side we have the root, and keep in mind that as those corn or soybeans or cover crop is growing, it's not only putting on biomass on top, but it's pumping carbon into the ground. Why is it willing to do that rather than put it towards more leaf area? Because it's donating or giving a lot of that carbon to microbes, which consume it, and in the process they release nitrogen, phosphorus, and other nutrients that the root can then absorb. So there's this pool of microbial and biota that's grazing on this carbon source. In the process of grazing, they're releasing these enzymes. And then at the same time, some of that nutrients and some of those organisms are being consumed and going up the food chain. So we have our nematode study to understand maybe the flow of nitrogen or other nutrients through the food web. And then some of that gets re-released again at the top of the food web and may get stabilized or end up on different fractions of the soil. So we can track some of the activity and the ways that we know that phosphorus is getting through the system. And then we can do, this is the part that takes longer that Jen just sort of mentioned, we can do a sort of really thorough analysis of different fractions of phosphorus. So we can extract phosphorus that's relatively easy to get, that a plant could get really easily, and then something that's a little more intermediately available. In chemical terms, that means, in chemical terms, that means we have to use something stronger to get it out of the soil, extract it more, and then we have a high acid to get out all the rest of the phosphorus. And that tells us where phosphorus is in the soil, where it's sort of stuck and building up, or where it might be moving. And then we can do that within the context of total phosphorus. So you've heard a lot about phosphorus in the water profile, acts differently in the soil. So we're trying to get an understanding of where phosphorus is moving within the soil and the processes by which it's being accessed. So this, in conjunction with the work of the Jeremiah Asher and the Water Institute. So the next phase of this will be to put edge of field monitoring for nutrients and phosphorus on at least 6 fields. And this will help us connect water quality on the fields or at the edge of the fields with what's happening within the fields with our soil health measures. And that will be able to measure surface and subsurface water. And we can supplement this data with IWR, with AGL, Limnotech data to get a fuller understanding of what's happening. So what's going to go in? This is sort of a mock-up of what the system will be. So these will be on fields with some kind of tile drainage. There's an independent power supply for an auto sampler and a weir installed. And then this auto sampler will periodically take at regular intervals a water sample, so we know what's actually ending up in the drain. And what that looks like is something like this, which will be installed. Here's the weir, here's the sampling port, and inside this box is the equipment that is being charged, the battery that's being charged, and all the equipment that's being used to measure a lot of the parameters that you saw earlier in the presentation. So all of that field, those field measurements or water measurements, hopefully add up to a big picture.
And because the water quality monitoring is so labor intensive, especially that edge of field piece, and we didn't have the funds to have a really large sample size here, we're also coupling that, harking back to the beginning here, the ecosystem approach to have a sense of nitrogen and phosphorus conservation or loss from all of the fields in the study. And we're doing that through this metric that's called a partial nutrient balance. And so this is basically, it's a simple measure in a way, but it requires a lot of detailed information from producers. So with those 5-year management histories, we can sum all of that up and get the total nitrogen or phosphorus input from all sources, fertilizer, legumes, manure, and then subtract out all of the nitrogen or phosphorus that's removed in harvested crops through time. And there's a really large literature now showing that sustained surpluses, so if you get a positive number over time, that's really well correlated with nutrient loss. This is, farms are ecosystems, they're subject to these laws of thermodynamics. This metric works well. And so this will give us just a sense of the trajectory and potential for nutrient conservation or loss for all of those management practices. And then we also will have the more detailed edge of field monitoring for a smaller subset along with the broader AGL network. So we don't have these data ready to share yet. We're still filling in some missing pieces from the interviews, and it also takes a little while to calculate these because we need to pull in various sources of information. We're getting near the end here. I'm just going to say a couple of things about next steps and then open it up for discussion. So in addition to the water quality monitoring, which will happen for the next 3 years, that's coupled with a second phase of this project, which is going to cover hopefully about half of the sample. So right now we have 28 fields that are enrolled in what we're calling phase 2. Those farmers have decided to implement a new conservation practice in those fields, everything from ramping up cover crops to cover crop mixtures, or trying no-till or reducing nutrient inputs. They're going to keep those practices in place over the next 3 growing seasons. I put this up because you just heard this overview from Brittany. This was a really important source of support for having some of the farmers stay in. And so we're going to stay in close communication with this group over the next 3 years and then resample in fall of 2027. And what that will do is allow us to understand change over time in soil health within the same field and with a known change in management. So that will really help us zoom in more on these indicators that are really appropriate for our region and more sensitive. We know, Tim Boring mentioned this morning, like it's just known that these practices take time to see the effects. 3 years isn't long enough for everything, but it'll be interesting to see which indicators are sensitive in that period. And then the last thing I'll say is just there is a community outreach component to all of this. And just as one piece of that, again, a shout out to Beth for while we're doing fieldwork and lab analyses, also whipping out these reports. We want to report back frequently to folks who are participating in the study. This is just a snapshot of a report that we're about to send out that has these preliminary results. So this is just an overall summary, and each farmer will get an image of where we sampled to refresh their memory, showing the locations of the samples. And then this is an example of the most complicated table. A lot of folks just had 1 or 2 fields sampled and not as many soil types, but they get individual results by field or soil type, the mean results for the study and the range across the study, along with the detailed glossary explaining what all of that means. But there will be much more to come. We're going to have even more interesting results hopefully coming out by December, and also try to include some basic things like potentially some high-level management recommendations or other types of interpretation. We're going to have regular roundtable meetings that we invite the participants to share these aggregate results and get feedback on interpretation and just what results mean to growers. We're also presenting at field days, other events, and again, trying to stay in close communication, because really our goal is to build these relationships and help support adoption of practices, but also really inform MDARD's conservation programs and where investments are going over time by sharing this feedback that we get from farmers in the broader community.